Welcome back, friends. As I promised you in the last video, that in the next video we shall be discussing about cell membrane. Stay tuned, as I told you before that cell membrane consists of many components and this discussion is slightly longer than the first one. However, because I said even in the cell wall the discussion will be slightly longer, you may think that this should be or will be shorter, like that of 10 minutes. No, this is longer. Now, cell membrane is an external thin structure enclosing a massive protoplasm. As I told you earlier, protoplasm is the combination of nucleus and the cytoplasm of a cell. Protoplasm is wet and running without some extraneous coating is rarely encountered. The cytoplasm of cell is surrounded by cell surface membrane. The cell surface membrane is the actual outermost protoplasmic layer of the cell. It is universal characteristics of all cells and it serves to separate and protect a cell from its surrounding environment. So this is the major function of the cell surface membrane. It serves to separate and protect. Separate a cell from outer environment and protect it. The cell surface membrane, also known as plasma membrane, or cytoplasmic membrane, or cell membrane, or plasmalemma, is believed to be formed by the cytoplasm as its own outer margin, and it is made mostly from a double layer of lipid, which are called phospholipids. So cell membrane has several names such as cytoplasmic membrane, plasma membrane, cell membrane, plasmodema, cell surface membrane. It's just the same, so don't confuse. And it is believed that it is formed from cytoplasm, inatengenezwa kutoka cytoplasm by the layer of phospholipids. Now, the cell membrane has been known longer and studied more intensively than internal cell membranes. Because cell membrane nje imekuwa studied more compared to internal cell membranes such as those of mitochondria and chloroplast. Third, the cell has many membranes besides the outer one. Cell ina membrane nyingi, sio tu ile nje. The membrane is said to be semi-permeable in that it can either let a substance or molecule or ion to pass through freely, pass through limited or not pass through at all. Kwa semi-permeable manake inaweza karuhusu kitu kipite free, kipite limited au kisipite kabisa. Now the cell membrane is able to control in and out movement of material because embedded in phospholipids by layer of the cell membrane, variety of proteins molecules that act as channels and pumps that move different molecules into and out of the cell. So cell membrane has different proteins incorporated in it, allowing different materials to pass in and out of the cell. Now in animal cell and some protozoans such as amoeba, it is an outer boundary, but in plant cell, fungal and bacteria. In plant cell, fungal cell, alga cell and bacterial cell, it is located beneath the cell wall. Other membranes include nuclear membrane or nuclear envelope. You have tonoplast surrounding the vacuole, which includes vacuole of plant cell, and the membrane of various cell organs such as endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, and lysosome. Now, structure of cell membrane. On the basis of structure, here we have two models or two hypotheses suggested by two different scientists. Kwa hiyo model ya kwanza inaitwa Daniel Davison model, ya pili fluid mosaic model. hizi na utofauti iliyoanza na Daniel Davison. According to Daniel Davison model, the membrane structure is made up of 2 layers, phospholipid layer and 2 protein layer. You must be very serious in order to understand this concept. A membrane is made up of 2 layers, phospholipid and 2 protein layers. But what did they say? Phospholipid bilayer is sandwiched between 2 protein layers. They said phospholipid bilayer is between, sandwiched means it is kept, we put it between. So it's sandwiched between 2 protein layers. A unit membrane had the protein, lipid, protein. Protein molecules layer is located outside the membrane. So that is the first idea of the Daniel Davison. So according to them, this is the protein and this is the protein. Then here is in between there is a phospholipid. Now this is what Daniel Davison, they suggested, that we have 2 protein layers and then in between we have phospholipids. Now protein molecule is located outside the membrane. Phospholipid molecules have got 2 parts, the polar head, which is hydrophilic, and the non-polar head, which is hydrophobic. The protein layer is continuous. Remember this word, is continuous, and the membrane therefore static and lack pores. So here is where these 2 scientists, Daniel and Davison, they failed to say membrane is continuous, lack pores, and static. The lipid are associated with phosphate group forming phospholipid bilayer. So issue of phospholipid ni kweli, issue of protein ni kweli, lakini swala kusema static, protein ni continuous na zimelaki pores ndio uongo. Now here they say each phospholipid molecule has a polar head, which is charged part. This part is polar and is water loving, hydrophilic. Hydrophilic ni water loving, kitu kinachopenda water. Where uncharged part is non-polar, is water hating, that is hydrophobic. Kwa hiyo according to Daniel Davison, structure cell membrane ilikuwa inaonekana kama protein, phospholipid core, protein coat. Kwa ukija kuangalia kwenye strength za Daniel Davison, this is the structure of cell membrane according to Daniel Davison. Ni iko hivyo. Kwa kwenye strength, it is true that membrane is chemically composed of protein and phospholipid bilayer. And it is also true that phospholipids have polar head and non-polar tail. These are true. But in the weakness of the Daniel Davison, it is not true that the membrane is static. The membrane is always over-changing or dynamic structure. The second thing, Daniel Davison failed to indicate the presence of pores in the protein layer in the membrane. So they said the protein is continuous, while it is not continuous. There are pores which allow different movement of molecules in us. It is not true that the protein layer is continuous without pores. Protein layer in the membrane is ideal, always not continuous, and there are pores within a protein molecules and between adjacent protein molecules. Kwa hiyo, these are 2 defects or weakness of the Daniel Davison model. Also, the third thing, it does not indicate the presence of carbohydrate, whereas membrane always contain carbohydrate. Kwa hakusema kwamba kwenye membrane kuna carbohydrate. Na 4, it does not indicate the presence of cholesterol. Now, due to those defects, there was development or suggestion of the new theory about the structure of cell membrane, and this was called the fluid mosaic model. Now, this model was proposed by J. Singer and Nicholson in 1972 to modify the Daniel Davison model. Kwa hiyo 1972 ndiko ambako Daniel Davison model liko modified. According to this model, the membrane has got 3 layers. Daniel Davison, they said 2, but this are three layers, namely lipid, protein, and carbohydrate. The layer of lipid, layer of protein, and layer of carbohydrate. The protein molecules, which act as mosaic, because this is called fluid mosaic. Lipid act as fluid, and protein act as mosaic. If you can't understand this, assume that you have a sea with the water. Then this sea with the water, there are boats. These boats, they are just rounding in the sea. Kwa ni kama tu ambavyo unaweza kaenda labda mahali fulani, kwa wale ambao labda ni wenyeji wa mikoa kama Dar es Salaam au mikoa mingine kando ya ziwa, ambavyo unaweza kaziona boti zinazunguka au zinakuepo kwenye maji. Ndio structure ya cell membrane ilivyo, kwamba hii hapa ndo fluid, ambao fluid imekuwa made up by nini? Phospholipids, na ndani wao kuna cholesterol. Lakini hizi hapa, hizi ndio mosaic. Hizi mosaic ndio proteins. Kwenye phospholipids zinaweza ka-attach carbohydrate, na kwenye proteins zinaweza ka-attach carbohydrate. Kwa huku zinaweza ka-attach carbohydrate zikatengeneza, huku kwenye proteins zikatengeneza compound ambayo ni complex of protein and carbohydrate, na huku zikatengeneza compound ambayo ni complex of carbohydrate and lipid, as you shall see later. Kwa huku itakuwa ni glycoprotein, huku ni glycolipid. Sasa all of the things we shall go to discuss later, but this is what you should understand. A mosaic is like a boat floating where the fluid is made up of phospholipid. Kwa hiyo tunachokisema ni kwamba the protein molecule which are mosaic are floating over surface of phospholipid barrier, which is fluid. Hence the name fluid mosaic model. The scattered protein molecule resemble a mosaic, but since the phospholipid barrier is fluid mosaic pattern, so they form a fluid mosaic pattern. According to this model, fluid mosaic model, the membrane is chemically composed of three layers: protein, lipid, and carbohydrate, which branches from protein and from lipid molecule. Now, protein molecules are arranged in a mosaic fashion, which float in a fluid barrier. The fluid mosaic model membrane confirms that the membrane is made up of phospholipid layers, carbohydrate molecules, which form branching from lipid and protein molecules. Protein does not form a continuous layer covering between sides of the membrane, as proposed by Davison. So these are the ideas of the fluid mosaic model. But idea number 4 here, the membrane is regarded as dynamic of a changing structure in each protein floating in the barrier of the lipids also move about. Kwa kama nilivyokuambia hapa kwamba hizi boti hizi ni kama zina move tu hapa kwenye maji. Kwa ni kama zinakuwa mosaic. Membrane is not static, it is dynamic, because these mosaic which are proteins, they just move over the phospholipid barrier. Membrane also consists of cholesterol, type of lipid which increase fluidity of the membrane. Wakati ya sita, the experimentation of viscosity suggests that a membrane is of fluid consistency and not at all that there is a considerable sideways movement of lipid and proteins within the membrane. Kwa hiyo kifupi kwa hivyo, and point number 7, due to the presence of fluidity and the mosaic arrangement of protein molecules, this mode of membrane structure is called fluid mosaic model. This model is found to be applied to all biological membranes in general, and it is seen as dynamic of a changing structure. Hypothesis number 8, there are hydrophilic pores polar pores which occur within a protein, ambazo proteins zinaitwa channel protein, or between adjacent protein. These pores allow passage of polar molecules such as glucose, water through the membrane, hydrophilic interior. So membrane kama membrane yenyewe ni phospholipid. Phospholipid ile pale kwa sababu ile lipid yenyewe ni hydrophobic. Sasa hydrophobic maana yake ni water hating. Kwa hiyo membrane kama isingekuwa na proteins yenyewe kama yenyewe kupitisha water ingekuwa very difficult thing. But the presence of protein allow the passage of hydrophilic. Philic means loving. Hydro water, hydrophilic are water loving molecules, while hydrophobic, they are water hating molecules. Now lipids na water ni vitu ambavyo havichangamani. So phospholipid bilayer the water hating molecule, but proteins allow the passage of hydrophilic molecules. Now fluid mosaic model is therefore a widely accepted model for structure of cell membrane. It is based on phospholipid bilayer which form framework of the membrane. Phospholipid bilayer is of the fluid protein and are randomly floating on it. The scattered protein resemble a mosaic, but since the phospholipid bilayer is fluid, the protein form fluid mosaic pattern. Now, let's go to discuss one component of the cell membrane and the function of each. The first component is membrane lipids. The great majority of lipids in the biological membrane is phospholipid and cholesterol. Phospholipid and cholesterol. The most common phospholipid in the membrane is phosphatidyl ethanolamine. So it's not so much important to understand this difficult name even to pronounce, but it's just important to know phospholipid. So phospholipid has hydrophilic water loving head and hydrophobic water hating tail. Now from the structure above, kwa sababu ukichora hiyo hapo, huku ni water loving, huku ni water hating. Kwa hiyo manake zitakavukaa hizo hapo, if this is our cell membrane, hizi head zitaangalia nje. Remember hii ni aqueous environment. Kwa ni kuna water huko. Kwa water loving zitaangalia kule. Na huku pia ni vile vile. Huku kwa sababu ni aqueous environment, kuna water. Kwa hapa katikati patakuwa ni kama empty space. Empty space huku hizi zitageukia kutail zitakuwa zinaangaliana hapo katikati. So from the structure above, phospholipids are amphipathic or amphiphilic compound. Amphiphilic or amphipathic manake ni kama zina different polarity. That are compounds with both water loving and water fearing. The large non-polar fatty acid part of the phospholipid do not dissolve or form reaction with water, but associate either with other fat material. The phosphorus containing region of the phospholipid is electrically charged, hence very hydrophilic, is able to interact with water. Kwa huku kuna phosphate ambao kunaweza kuinteract na water, wakati huku kwenye tail hakuwezi kuinteract na water. That the phosphorus containing region of the phospholipid is polar charged and is known as polar head, while the two fatty acids of the phospholipid are non-polar or non-charged and are known as non-polar tail. Now the cell membrane is phospholipid bilayer, so because it is made up of two layers of phospholipid molecules. The phospholipid bilayer membrane is formed when the fatty acid of the two layers are towards the center and the polar head and charged region facing outside separating two aqueous water region, kwamba head zinakuwa zina face outside and the tail they are facing together is nilivyokuwa nime illustrate kwa diagram. When the phospholipids are exposed to water, they arrange themselves into two layered sheets, a bilayer, with all their tails pointing towards the center of the sheet. The center of this bilayer contains almost no water and excludes molecules like sugar or salt that dissolve in water, but not in oil. Certain kind of membrane protein are involved in the process of fusing two layers together. Cholesterol, which help to strengthen the bilayer and decrease its permeability, also help to regulate activity of certain integral proteins. Now the rule of proteins. Ni maswali ambayo wanauliza. Rule of lipids. The phospholipid portion of the membrane stabilizes the entire structure and gives the membrane fluidity. Phospholipids enable passage of fat-soluble substance across the membrane. Phospholipids prevent the membrane from solidifying at a very low temperature, which may result into the bursting of the cell membrane. Kwa hiyo naona phospholipid zinasaidia katika kustabilize entire structure. Phospholipid zina enable passage ya fat soluble molecule, lakini pia zina prevent solidification of the membrane at low temperature. Lakini ya nne, because of the tendency of fatty acid to associate with one another and exclude water, small holes or rips in the membrane seal them spontaneously. This facilitates the fusion of membrane associated with endocytosis, exocytosis and cell fusion. Kwa pia zinasaidia katika process hapo endocytosis, exocytosis na cell fusion. Ya tano, hydrophilic head of the phospholipids are pointed towards out of the phospholipid bilayer. This enables the cell to be in contact with water on both sides. Kwa cell inakuwa ina water kwa sababu hydrophilic head zimekuwa in contact with water. The sita, protein offers structural support of the membrane. Protein manake ambazo zimekuwa associated katika phospholipid. A saba, protein associated with carbohydrate to form glycoprotein, which also may act as receptor sites of the hydroxyl group of the nature of the cell membrane. Now from there, features. Tumeshaona hiyo lipidi. Features of the mosaic model of the membrane structure. Features. Hizi features ziko kwenye BSc na huwa zinaulizwa. Utaje features of the fluid mosaic model. Ya kwanza, it is a thin structure about 7 micrometer thick. You have to claim them as they are. Ya pili, the basic structure is a phospholipid bilayer. Ya tatu, the hydrophilic phosphate heads of the phospholipid face outward into the aqueous environment, inside and outside this. Ya nne, the hydrocarbon tail face inward and create a hydrophobic interior. Ya tano, the phospholipids are fluid and move about rapidly by diffusion in their own layer. Ya sita, some of the fatty acid tail are saturated and some are unsaturated. Unsaturated tail are bent and fit together more closely. Therefore, the more unsaturated the tail are, the more fluid the membrane is. Ya saba, most protein molecules float about in the phospholipid layer forming a fluid mosaic pattern. Ya nane, the proteins stay in the membrane because they have region of hydrophobic amino acid which interact with the fatty acid tail to exclude water. Ya tisa, some protein penetrates only part of the way into the membrane while other penetrate all the way through. Ya kumi, some protein and lipid have short branching carbohydrate chain like antenna forming glycoprotein and glycolipid respectively. Ya 11, membranes also contain cholesterol like unsaturated fatty acid. Cholesterol disturbs the close packing of the phospholipid and keep them more fluid. This can be important for organisms living at low temperatures. When membranes can solidify, cholesterol also increases flexibility and stability of membranes, without it membranes break up. Ya 12, the two sides of the membrane can define composition and function. Kwa hiyo kuhusu diagram hapo, hii ni figure 5.16 kwenye BS page namba 142. Pia kuna swali 5.1 kwenye BS na lina majibu yake pia kwenye BS umeona. Kwa hiyo ni hapa sina BS, ningeweza kuwasomea, lakini I don't have BS here, I don't have access to BS. Sasa all about the cases of membrane lipids tumeshaziona. Lipids constitute 45% of the total chemistry of the cell membrane. There are 3 types of lipid: phospholipid, glycolipid, and cholesterol. Phospholipid manake lipid associated with phosphate, glycol associated with carbohydrate, and cholesterol is on steroids. Tanda kuona kwenye biochemistry topic 2. So phospholipids, there are 2 layers of phospholipid molecule, hence phospholipid bilayer. They exist as fluid in the membrane and they are always in constant motion. Phospholipids make the basic structure of the membrane. They act as framework since they are in fluid state. They help to determine the fluid nature of the membrane. Phospholipids make the outer and inner layer of the membrane, and in this acts as a barrier to the entrance of some molecule. Phospholipids therefore affect the fluidity and the permeability of the membrane. Kwa hiyo ni phospholipids. Hapo utakuta kuna functions of phospholipid: provide basic structure of the membrane, determine fluidity of the membrane. 3, allow passage of soluble substance. Kuna glycolipid. These are formed when carbohydrate molecule attach on the lipid molecule, in this case phospholipid. In other words, glycolipid is formed when carbohydrate molecule attach on lipid molecule. Glycolipids are found only on the external surface of the cell membrane, not in the internal. Function of glycolipid act as a recognition site or receptor for recognition of the external stimuli. Kwa unavyosikia stimulus fulani imekuwa received by a cell, the function of glycolipid. Ya pili, it helps to make the membrane more stable, or it strengthens the membrane. Ya 3 ni cholesterol. Cholesterol are lipid steroids which are embedded in the phospholipid bilayer. They make the phospholipids bilayer stronger and more flexible. They make the membrane less fluid at high temperature and more fluid at low temperature. So when the temperature is high, cholesterol, they make the membrane less fluid, and when the temperature is low, they make the membrane more fluid. Another thing, they make the membrane less permeable to water-soluble substances such as glucose. Another thing, they maintain the fluidity of the membrane. Kwa hiyo ni hivyo tu. As they kept a part of phospholipid molecule, cholesterol disturb the packing of phospholipid. Now, what would happen in the cell if we couldn't have cholesterol? Ni maswali ambayo unaweza kuulizwa direct. What would happen in the cell membrane if we couldn't have cholesterol? Now, in the absence of cholesterol, the membrane would be too fluid and would burst just like soap bubble at high temperature. Kwa at high temperature, membrane inge burst kama soap bubble. Ambacho ni kitu tumesema cholesterol kwenye high temperature inafanya membrane iwe more rigid. A pili, membrane would solidify and collapse at low temperature. So this based on the function of cholesterol to regulate the fluidity of the membrane. Now, membrane proteins. Protein consists of about 45% of the total chemical substance of the membrane, embedded on or within the phospholipid bilayer of the membrane. There are a variety of protein molecules that act as channels and pumps to move different materials into and out of the cell. 2 types of protein according to location are found in the cell membrane. Kwa kuna intrinsic and extrinsic. Now, Intrinsic protein. These are proteins which penetrate into the phospholipid bilayer and may even extend from one side to the other. They extend from one side of the membrane to the other. The proportion of the protein molecule that reside within the lipid bilayer is very hydrophobic, has high content of hydrophobic amino acid. Kwa ile proportion ambayo inakaa ndani ya phospholipid bilayer ina content kubwa ya hydrophobic amino acids. Lakini pia extrinsic membrane protein. These are proteins located on the outside of the phospholipid bilayer and are attached to the rest of the membrane by weak non-covalent bonding. So they are located outside of the phospholipid bilayer and are attached to the rest of the membrane by weak non-covalent bonding with the intrinsic protein or hydrophilic amino acid. Kwa hizi zinakuwa just only a portion. Kama tungechora manake tungesema if this is the membrane which is phospholipid bilayer, and let's say here we have another membrane, so intrinsic protein zina uwezo kutoka huko nje, while extrinsic protein, so both of them they are floating on the cell membrane, but the difference is that the intrinsic protein they can pass from one side to the another, while the extrinsic, manake hapa ndo tunkuja kuikuta phospholipids mzimu, extrinsic hazifanyi hivyo, kwa ndiye tunkuta phospholipids. Kwa hiyo vitu viko kama hivyo. Note, membrane proteins are free to move laterally through the lipid bilayer. Thus, the lipid bilayer is especially a film of oil. Structures immersed in it would be relatively free to float about it. Kwa ni kama vile tu ambavyo unaweza kuona kama vile ukiweka mafuta afu umeviweka vitu fulani juu yake zina float. Also, protein composition of the membrane of various organelles differ because the membrane perform different biochemical reactions such as respiration, photosynthesis, etc. Also, lipids and protein found in the cell membrane differ from those found in the internal membranes. Lastly, some proteins may be confined to one part of the cell surface rather than scattered evenly about. According to fluid Mosaic model, the protein layer is not continuous. It is collection of protein globules that float over the phospholipid bilayer. Now, rules of protein. Protein maintains structure of the cell membrane. Proteins act as enzymes for some reaction. Act as recognition site. That act as receptor site for chemical signals. Lakini pia proteins act as receptor molecules ambayo tushaelezea. They act as energy transducer and electron carrier. Unaona? Protein they form channel or pathway through which material pass across the cell membrane. Kutoka ndani kwenda nje na kutoka nje kuingia ndani. Lakini pia proteins such as antigens act as cell identity maker. Kwa hiyo cell inakuwa na protein fulani kwenye cell membrane ambayo tunaweza kujua ile cell kwa ni cell fulani kwa sababu ya antigen yake. Ya saba, some protein catalyzes chemical reaction in the membrane surface. They are not enzymes, they are enzymes, but they are enzyme confined on the surfaces of membrane. They are not released. Ya nane, protein associated with carbohydrate to form glycoprotein, which also may act as a receptor. Now, the last but not least component of the cell membrane is carbohydrate. The carbohydrates are attached to either lipid or protein molecule, and they are confined to outside with the plasma membrane, where they protrude into the cell environment. Now, carbohydrates branch from lipid phospholipids. and protein forming glycolipids. Kwa inaweza kuwa glycolipid au inaweza kuwa ni glycoprotein. They branch like antenna which extend outside the membrane. They act as recognition site for receptor. Loss of carbohydrate. Loss of carbohydrate in the membrane. Carbohydrates are important in all sorts of recognition reaction to the cell surface. Carbohydrates recognize site of origin substance at the cell. Carbohydrates act as receptor sites in the cell membrane. Strength of fluid mosaic model. It is true that the membrane is made up of protein, lipid, and carbohydrate. It is true that protein layer is not continuous. It indicates the presence of pores in the membrane. It indicates that the membrane is not static, rather than it is dynamic or even changing structure. Now, general functions of the cell membrane. The first one is to control materials entering and leaving the cytoplasm or passage of material. As we said that the cell surface membrane is selectively permeable. The second one, it acts as a receptor site for external changes stimulus. Proteins have very specific shapes. This makes them ideal as a receptor molecule for chemical signal between cells. Ya tatu, it separates cytoplasm and its inclusion from external environment. Separate control the cell. Yani it allows interaction with other cell. Internal cell membrane makes separate compartment inside the cell which specialize it in specific metabolic pathway, e.g. photosynthesis in chloroplast, respiration, mitochondria. A system acts as the site of occurrence of different metabolic reaction. Kwa mfano, light reaction of photosynthesis in take place kwenye membranes. Ya saba, cholesterol act like a plug, reducing even further the escape or entry of polar molecule. Ya nane, channel proteins and carrier proteins. These are involved in selective transport of polar molecule and the ions across the membrane. From there, tisa, it is used in energy transfer system in photosynthesis and respiration that exists in the membrane of chloroplast and mitochondria. Kwa inatumika katika energy transfer system hiyo tutaenda kuiona kwenye photosynthesis au kwenye respiration. Ya 10, glycolipid are branching carbohydrate side chain and they are involved in the cell recognition. Ya 11 ni antigens ambazo ziko kwenye cell membrane. Lakini pia 12 ni enzymes. Antigens zinatumika katika cell recognition na enzymes zinatumika katika different processes of the cell. Now here there are some of the questions you can use to measure our understanding, to measure to how much extent we have understood. The first, name the major chemical constituent of the cell membrane, structure and state the function of each. Kwa hiyo major hapo maana kitakuwa ni protein, lipid, carbohydrate. State major function of each. Year P, discuss the structure of the membrane of Singer and Nicholson model and point out its strength and weakness. Singer and Nicholson means it is fluid mosaic model. Number 3, the diagram below shows a section of cell membrane. Study it carefully and answer the questions. Kwa kutoka kuna A, kuna B, kuna C, kuna D, E, F, G. Kwa mfano swali la kwanza lilikuwa na kuhitaji ufanyaje ulebo hizo. Ulebo A to H. A is hydrophilic AD, hydrophobic tail, phospholipid, phospholipid barrier, protein channel, cholesterol, glycoprotein, glycolipid. This is glycolipid. This is glycoprotein attached to protein attached to lipid. Kwa mfano swali A. Swali B, fluid mosaic model of the membrane is described as dynamic what does dynamic imply in this context? Ndicho kitu ambacho tulisema kwamba the proteins always move around, or they are floating over the mosaic. So that's what the term dynamic means. List 4 functions of the cell membrane protein. Give 2 functions of glycoprotein in the membrane. Briefly explain why cell surface membrane are described as having fluid mosaic structure. Cell membrane discussion, it has ended up here. Let us look maybe if there will be any of the addition in my another notes. Let us look if there will be any of the addition to add, increasing your understanding and your performance in exams. Okay. Anyway, ni kama vitu ambavyo tulishavisema, lakini tuvidiscuss tena. Ni adaptation of cell membrane to its function. Ya kwanza, it is thickness, yani ni very thin, which allow easier and faster movement of substance across it. Ya pili, it is pores, which allow the passage of hydrophilic polar molecules. Ya tatu, it is lipid in nature, and the possession of pores make it partially thus enabled to control what interleave the cell. Ya nne, has cognizing site, they form glycolipid and glycoprotein. Ya tano, has cholesterol, which is stable in both high and low temperature. Ya sita, has protein which act as enzyme. Ya saba, dynamic, versatile, and even changing. This is changing in order to perform a number of different functions. Next lecture, we shall start to look on the protoplasm, that is cytoplasm and organelles. We shall start looking one organelle after another, and I hope we shall start with nucleus. We shall start with the double membrane organelles such as nucleus, chloroplast, mitochondria. Then we shall go to other organelles, and it is where we shall finish cytology, but it's not easier. It is still a long journey. Wish you nice studies. Subscribe my channel, join my Telegram group for more and more updates. If you have any question you have not understood any part, just comment down below, and then I will work with your comment.